Welcome, students, to our video notes on 10.2 and 10.3, collecting data. We're going to talk about the difference between qualitative and quantitative data, populations and samples, parameters, statistics, bias, interpret relative frequency, and distinguish between bar charts and pie charts. We're going to look at histograms, box plots, the shape of different distributions, and how to use shape, center, and spread. A lot of this you've seen before. But we're going to go over these definitions because the vocabulary is super important. So statistics is the idea that we don't have all the information about a population, and so we're going to look at a small subset that we do have information about and try to gather inferences or assumptions about the entire population. Observational data is when we collect something without influencing the responses. So there's no treatment. We're just observing something that's already happening. We're asking questions about people's current habits. Designed experiments are where we impose a treatment on experimental units or subjects. Univariate data, uni means one. So one, variate means variable. So one variable data only have a single character. Qualitative data is in categories. It's also called categorical. So that would be like eye color or jersey number or address. Quantitative data is numerical. So that's something you can measure and something that it makes sense to take an average of. So that would be like temperature or height. When we sample, we look at the entire population, and usually we don't have enough information about everybody. So we're trying to estimate some parameter, some fact about a large group of people. So what we do is we take a sample, a smaller portion of that population, and we collect whatever data we were interested in about that little group, and that's called a statistic. So if we want to know the height of the whole population, we look, which is a parameter, we look at the height of a sample, which is called a statistic. Simple random sampling is where we take all of the individuals in a population and we randomly assign them, or we assign them numbers, and we randomly choose a portion of those to sample. Bias is a result when samples are not representative. Raw data is data that has been collected but not organized or summarized. The frequency is the number of times an event occurs divided by the number of trials. A bar graph and a circle graph, or a pie chart, more commonly known, are the best ways to organize and summarize categorical or qualitative data. So these are for things like eye color and preference and yes or no questions. Quantitative data, on the other hand, requires histograms or box plots. Histograms, as you can see, show the data more linearly. Box plot is a summary based on the 5-number summary. 5-number summary consists of the minimum, quartile 1, the median, quartile 3, and the maximum. So the median is obtained by ordering all of the numbers from least to greatest and then chopping off the lowest and the highest and the lowest and the highest until you get to exactly the middle number. Quartile 1 is taken by taking the median and the minimum and doing that same thing, chopping the median, chopping the minimum until you get to the middle of the first half. Quartile 3 is the middle number of the top half. The maximum is the highest number, the minimum is the lowest number. The interquartile range is what we get by subtracting quartile 3, Q3, by Q1, which is quartile 1. And that's the width of the box in a box plot. Thank you so much for watching.